are going with the VSAQs. Already in the question paper, recent question paper 2023 board exam paper, whatever VSAQs have been given, I have taught in the previous session. And even in yesterday's session, that is Tuesday, yesterday's session also, I have started the important VSAQ from the first chapter. So digestion and absorption, breathing and exchange of gases, body fluids and circulation. From all these chapters, already we have completed important VSAQs. So today, let us go with remaining VSAQs, very short answer questions of the other chapters. So let us see important VSAQs of excretory products and their elimination. So in that first one is, what are columns of Bertini or renal columns? What are columns of Bertini or renal columns? This is the question. So what are columns of Bertini or renal columns? So what is the answer which you should write? In kidney, the projections of the cortex. So in kidney, the projections of the cortex that which are extending, so that which are extending in between the renal pyramids, in between the medullary pyramids or the renal pyramids. So in kidney, the projections of the renal cortex between the renal pyramids and kidney, renal pyramids in kidney. See, they are only called as what? Columns of Bertini. So what are columns of Bertini? In kidney, the projections of the cortex that which are extending in between the renal pyramids. So for example, if you take the kidney, so you know that, so the kidney, it contains many renal medullary pyramids like this, right? So like this, the renal pyramids are present that which are actually opening into the calyces. The projections called as calyces, again, that which unite together. Okay, so like minor calyces, major calyces, so like this, they would be present. So now what happens? So these spaces, so the projections, these projections of the renal cortex that which are extending in between the renal pyramids. So these are called as renal pyramids. So in between the renal pyramids, there are extensions of what? There are extensions of the cortex. So that's why they are called as what? So these projections are only called as columns of Bertini. So these projections are only called as what? Columns of Bertini. So this is important question. What are columns of Bertini? So in kidney, the projections of the cortex that which are extending in between the renal pyramids of kidney, in between the renal pyramids of kidney. So they are only called as columns of Bertini or renal columns. So which is very important question, 2-mark question. So then next in excretory products and their elimination only, let us go with the other important question. So that is define glomerular filtration. Another important question is define glomerular filtration. Actually, here 2 questions are there. Glomerular filtration and glomerular filtration rate. So glomerular filtration and glomerular filtration rate, both questions are there. So both are important only. So what is meant by glomerular filtration? So glomerular filtration is the first step in urine formation, which is a passive process. So glomerular filtration is the first step in urine formation. Actually, urine formation, it involves 3 steps. Glomerular filtration, tubular reabsorption, and then tubular secretion. So in that, glomerular filtration is the first step in urine formation, where it is a passive process. So it is a passive process. And what is this? The filtration of blood. The filtration of blood. The definition for this is the filtration of blood from the glomerulus. The filtration of blood from the glomerulus into the lumen of the Bowman's capsule. into the lumen of Bowman's capsule. The filtration of blood from the glomerulus into the lumen of Bowman's capsule is only called as glomerular filtration. So 2 points you should write. One is definition and the other one is, no, it is which step of urine formation you are supposed to write. So what is the definition? The filtration of blood from the glomerulus into the lumen of Bowman's capsule is only called as glomerular filtration, which is the first step of urine formation, and it is a passive process. That means energy is not involved for this. And see, for example, you know that Malpighian body, it consists of Bowman's capsule, the cup-shaped Bowman's capsule, and inside it contains network of capillaries, where these capillaries are formed by means of afferent arteriole, and again that which combine together to form efferent arteriole. So now the filtration of blood, so from the glomerulus into the lumen of this Bowman's capsule, so into the lumen of the Bowman's capsule. So this is only called as what? Glomerular filtration. The next one more question is there. Define glomerular filtration rate. This already we have discussed even in the previous session. So just let me revise once. Define glomerular filtration rate, GFR. GFR. So third question is define GFR. Glomerular filtration rate. So you have understood what is glomerular filtration. That is filtration of blood from the glomerulus into the lumen of the Bowman's capsule is only called as glomerular filtration. Then what is this GFR? The question itself indicates that it is the rate. It is talking about the rate. So the rate at which the amount of filtrate, so the rate at which the amount of filtrate is formed by, so the amount of filtrate which is formed by all the nephrons present in both the kidneys, by all the nephrons present in both the kidneys per minute, is only called as glomerular filtration rate. So that is only called as GFR, glomerular filtration rate, GFR. The rate at which the amount of filtrate which is formed by all the nephrons present in both the kidneys per minute is only called as GFR. So that is equal to how much? 125 ml per minute or 180 liter per day. So it is 125 ml per minute or 180 liter per day. So this is the difference between glomerular filtration and glomerular filtration rate. So glomerular filtration. So what is glomerular filtration? You are supposed to write like filtration of blood from the glomerulus, and glomerular filtration rate is how much amount of filtrate is formed by both the kidneys per minute. So that is glomerular filtration rate. So that is the difference between the two. So totally 3 questions we have discussed till now. Columns of Bertini, glomerular filtration and glomerular filtration rate. So these 3 are very, very important. And the next important question, distinguish between the terms renin and renin. So actually 2 things are there. One is renin, R-E-N-I-N, and the other one is R-E-N-N-I-N. So this question also important. Distinguish between the terms renin and renin. R-E-N-I-N, that is related to kidney, and R-E-N-N-I-N, so that is related to digestion. So like that you should remember. So fourth question, distinguish between the terms renin and renin. Hope that you got the difference. R-E-N-I-N and R-E-N-N-I-N. First of all, let us see what is R-E-N-I-N, renin. So renin, it is what? It is actually produced by means of JGA cells, juxtaglomerular cells of the kidney. So juxtaglomerular cells of the kidney, which is an enzyme. It is an enzyme. So it is an enzyme which is secreted by JG cells of kidney, juxtaglomerular cells of the kidney, where this is involved. It is involved in the conversion of, what? Angiotensinogen, inactive angiotensinogen, to angiotensin 1. Angiotensin 1. Okay. So renin is mainly involved in the conversion of angiotensinogen to angiotensin. Angiotensin 1. And again, actually, angiotensin 1 is also converted into angiotensin 2 by means of angiotensin converting enzyme. Okay, angiotensin converting enzyme. So now whatever this angiotensin 1 is formed, that is converted into what? Angiotensin 2 by means of ACE. What is ACE? Angiotensin converting enzyme. Now what this angiotensin 2 will do? It is a powerful vasoconstrictor. So thereby it will constrict the blood vessels and increase the blood pressure. And not only that, even it will go and stimulate adrenal cortex to secrete aldosterone. It will go and stimulate adrenal cortex to secrete what? Aldosterone. So thereby aldosterone will cause active reabsorption of sodium, thereby again increasing the glomerular blood filtration rate. So thereby GFR rate will be increased. So like this, renin is mainly involved in the conversion of angiotensinogen to angiotensin 1, that which is required in order to increase the GFR, glomerular filtration rate, or the glomerular blood flow. Okay. So mainly renin is involved for that. Then what about renin? RENNIN. So renin, it's a proteolytic enzyme. Proteolytic means what? It is involved in breaking of proteins. And this renin, it is actually secreted from gastric glands of the stomach. Secreted from gastric glands, the two chief cells of stomach. So renin is mainly secreted from the gastric glands of the stomach in infants only. Particularly this enzyme is secreted in infants. So whenever the infants are growing, what happens? Slowly the secretion of this renin also slows down and finally it gets stopped. So this renin, it is mainly involved in the conversion of what? It is mainly involved in the conversion of milk protein casein. Milk protein casein to paracasein. Milk protein casein to paracasein, where this paracasein again combines with calcium ions to form calcium paracaseinate. Which is in curdled form. So that is the reason why infants, after drinking milk, if they vomit after few minutes like that, so we can see that whatever the milk they have taken, that coming out in the form of a curd, in curdled form, right? So the reason for that is action of renin on casein. So casein is a milk protein which is present in the milk, where the renin is acting on the casein and converts that into paracasein. And paracasein, by combining with calcium, converts that into calcium paracaseinate. So like this, no? There are two terms, renin and renin. RENIN and RENNIN. So what is RENIN? Renin. So it is produced, it is an enzyme which is secreted by JG cells of kidney, where it is involved in the conversion of inactive angiotensinogen to angiotensin 1. And angiotensin 1 again in the presence of ACE, angiotensin converting enzyme, it is converted into angiotensin 2. At least if you remember these two reactions and write also, you will get at least 1 mark. So that's why learn at least the reactions. Then renin is a proteolytic enzyme secreted from gastric glands of the stomach only in infants. And this renin, it acts on casein and converts that into paracasein. And paracasein, by combining with calcium ions, so gets converted into calcium paracaseinate. So this is the difference between the enzymes renin and renin. The next, coming to the differences between columns of Bertini and ducts of Bellini. Next question, what are the differences between columns of Bertini and ducts of Bellini? That is also important, but not that much important. But still, if you have time, you can learn that question also. But till now, whatever I said, all these are very important. Glomerular filtration rate, then columns of Bertini, and then distinguish between the terms renin and then renin. So now, next one, differences between columns of Bertini and ducts of Bellini. Already we have learned what is columns of Bertini. You should know what is ducts of Bellini. So just now we have learned that columns of Bertini is, so columns of Bertini and ducts of Bellini, you should write the difference between the two. So what is columns of Bertini? The projections of renal cortex, extending in between the renal pyramids of kidney. Extending in between the renal pyramids of kidney. So projections of renal cortex, that which are extending in between the renal pyramids of kidney, is only called as columns of Bertini. This is the first point. And what is this ducts of Bellini? Here collecting ducts of, that which are coming from different nephrons. So the collecting ducts of many nephrons, they unite together to form what? Ducts of Bellini. So ducts of Bellini is about what? Collecting ducts. So collecting ducts of different nephrons or many nephrons unite together to form a duct. So to form what? Ducts of Bellini. So collecting ducts of different nephrons, they unite together in order to form larger ducts, okay, which is called as ducts of Bellini. So in order to form larger ducts, opening into medulla, larger ducts opening into medulla of kidney. So opening into medulla of kidney, so called as what? Called as ducts of Bellini. So what is ducts of Bellini? Collecting ducts of different nephrons unite together to form larger ducts, that which are opening into the medulla of kidney. So they are only called as ducts of Bellini. So this is the difference between columns of Bertini and ducts of Bellini. And actually in columns of Bertini, what are present? Blood vessels, urinary tubes, and fibrous material are present in each column. So blood vessels, urinary tubes, and fibrous material are present in each column. So what are present in columns of Bertini? Urinary tubes, blood vessels, and fibrous material are entering into each column of Bertini. Okay. Then what is this ducts of Bellini? So the distal portion of this collecting duct system, it represents these ducts, and actually they are opening into the calyces, that which leads to pelvis. So now all these ducts of Bellini, they open into calyces. Okay, so that again opens into what? Pelvis, pelvic region of the kidney. So that again opens into the pelvis of the kidney. So ducts of Bellini, they open into calyces, that which again opens into pelvis, or that which leads to pelvis, and the distal portion of the collecting duct system represent these ducts. So the distal portion of the collecting duct system will represent ducts of Bellini. Clear? So like this, you should write the differences between columns of Bertini and ducts of Bellini. So what are columns of Bertini? Projections of renal cortex extending in between the renal pyramids of kidney is called as columns of Bertini. And what are ducts of Bellini? Collecting ducts of different nephrons. They unite together in order to form larger ducts opening into the medulla of kidney. And here in columns of Bertini, what are present? Urinary tubes, blood vessels, and fibrous material are present in this region or in each column. And whereas ducts of Bellini, they open into calyces, that which leads into pelvis. And the distal portion of the collecting duct system will represent this ducts of Bellini. So like this, you are supposed to remember the differences between columns of Bertini and ducts of Bellini. So totally 5 important questions I have told you in excretory products and their elimination. So then next, let us go with VSAQs of, that is musculoskeletal system chapter. Important VSAQs of musculoskeletal system. Important VSAQs of musculoskeletal system. In that, first question is, what is a triad system? So already today's session, I have given 5 important VSAQs. The next one from musculoskeletal system is, what is triad system? Which is very important question. What is triad system? So triad system is particularly in case of skeletal muscle. In skeletal muscle, so the T tubule is flanked. So the T tubule is flanked on its either side, by the terminal cistae of sarcoplasmic reticulum. By the terminal cisternae of sarcoplasmic reticulum. So sarcoplasmic reticulum is endoplasmic reticulum only, which is present in the muscle. So generally the endoplasmic reticulum of muscle is called as what? Sarcoplasmic reticulum. So triad system is, in skeletal muscle, the T tubule is flanked on its either side by what? The terminal cisternae of the sarcoplasmic reticulum. So now the T tubule and terminal cisternae. The T tubule and 2 terminal cisternae together. So T tubule is 1 and 2 terminal cisternae on either side of it together is only called as what? So that is only called as triad system. So that is only called as triad system. So in case of skeletal muscle, the T tubule is flanked on its either side by the 2 terminal cisternae of sarcoplasmic reticulum. Now the T tubule and 2 terminal cisternae which are present together. So that is only called as what? Triad system. That is only called as triad system. In case of skeletal muscle, the T tubule. So for example, if this is T tubule, so the T tubule, whatever is present and that which is flanked on its either side by the terminal cisternae of the sarcoplasmic reticulum, So now together that can be called as what? That can be called as triad system. So this is terminal cisternae, which are flanking on its either side. So now together it can be called as what? That can be called as a triad system. So then next, coming to another important question in musculoskeletal system. Already this question, during the question paper discussion also I have told you. Once again, let me revise. Name the keystone bone of the cranium and where it is located. Name the keystone bone of the cranium. Name the keystone bone of the cranium and where is it located. So name the keystone bone of the cranium and where is it located. So the keystone bone of the cranium is sphenoid bone. So sphenoid bone is the keystone bone of the cranium. Sphenoid bone is the keystone bone of the cranium. Already I told you that cranium contains totally 8 bones. Parietal bone, frontal bone, temporal bone, occipital bone, sphenoid bone. So like the totally 8 are there. In that 8, sphenoid bone is the keystone bone of the cranium and it is located at the base of the skull. It is located at the base of the skull. It is located at the base of the skull. So sphenoid bone is the keystone bone of the cranium, which is located at the base of the skull. So for these 2 points, you will get 2 marks. One is sphenoid bone and location is base of the skull. So these 2 are very important questions in musculoskeletal system. Apart from that, even we have discussed about the differences between actin and myosin, which is another important. So differences between actin and myosin, this question also already in the previous session, like in the question paper, I have discussed it with you. So actin is a thin filament, whereas myosin is a thick filament. So actin is present in light band, myosin is present in dark band. So in actin, the filaments are connected so to the Z line, and whereas in myosin, the filaments are connected to the M line. So like that already we have discussed the differences between actin and myosin. So once again, I am repeating the differences between actin and myosin. If you take, actin is a thin filament, I told you, and whereas myosin is thick filament. So actin is present in light band, whereas myosin is present in what? It is present in dark band. And in actin, the filaments are connected to Z line. The filaments are connected to Z line. And whereas in myosin, the filaments are connected to, or the filaments are in connection with M line. So that is membranous line. So, or connected to M line, membranous line. So like this already we have discussed the differences between actin and myosin. So totally 3 important questions in musculoskeletal system, differences between actin and myosin. What is triad system? Name the keystone bone of the cranium and where is it located? And even let us go with one more question. 2 more also let us discuss. That is the differences between red muscle fibers and white muscle fibers. So in this chapter, let us go with the 2 more important questions. 2 more important questions let us discuss. Hope that you have written these answers. Hope that you have written these answers. So let us go with another important question. So that is differences between red muscle fibers and white muscle fibers. So next question. differences between red muscle fibers and white muscle fibers. So what are the differences? Red muscle fibers, why they are red in color? And white muscle fibers, why they are white in color? So red muscle fibers, they contain higher amount of myoglobin. So red muscle fibers contain high amount of myoglobin, which is a red colored pigment present in the muscle, acting as a reservoir of oxygen. In Cori cycle also I told you. So red muscle fibers, they contain high amount of myoglobin. That is the reason why. So this myoglobin is what? A red colored pigment. It is a red colored pigment present in some of the muscle fibers. So the muscle fibers that which contain high amount of myoglobin, so they are called as red muscle fibers. And white muscle fibers. So what are white muscle fibers? White muscle fibers are those which contain white muscle fibers are those which contain low amount of myoglobin or less amount of myoglobin. That's why they are white in color. So they contain low amount of myoglobin. They contain low amount of myoglobin. And next one, in red muscle fibers, mitochondria are more in number. So thereby, higher energy will be released. So mitochondria are more in number. And whereas in case of white muscle fibers, mitochondria are less in number. Mitochondria are less in number. So here mitochondria are more in number and here mitochondria are less in number. Then in red muscle fibers, they are also called as aerobic muscles because these muscles mainly they will work whenever continuous oxygen supply is there. So that's why they are called as aerobic muscles. They are called as aerobic muscles. And whereas white muscle fibers, they are called as anaerobic muscles. So they are aerobic muscles and whereas these are called as anaerobic muscles. Aerobic muscles and anaerobic muscles. So red muscle fibers contain high amount of myoglobin. White muscle fibers contain low amount of myoglobin. In red muscle fibers, mitochondria are more in number, whereas in white muscle fibers, mitochondria are less in number. And red muscle fibers, they are called as aerobic muscles. And whereas white muscle fibers, they are called as anaerobic muscles. So these are the differences between red and white muscle fibers, which is important. At the same time, easy question. Very easily you can remember. Very easily you can remember this. Differences between red and white muscle fibers. The next coming to another important VSAQ from this chapter, last one. So that is, name the middle ear ossicles and their evolutionary origin. Name the middle ear ossicles and what is their evolutionary origin. So that is the question. Name the middle ear ossicles and what is their evolutionary origin. VSAQ in musculoskeletal system chapter only. So name the middle ear ossicles and name their and what is their evolutionary origin. So the three tiny bones which are present in the middle ear. So actually our human ear is divided into three parts. external ear, middle ear, and then internal ear. So in that middle ear, so the three tiny bones present in the middle ear, the three tiny bones present in the middle ear. So they are only called as what? They are only called as ear ossicles or middle ear bones. So the three tiny bones which are present in the middle ear are only called as ear ossicles or middle ear bones. So what are they? Malleus, incus, and stapes. Malleus, incus, and stapes. Malleus, the name has come because it is hammer shaped, and incus, it is anvil, and stapes, it is stirrup. So malleus, incus, stapes. Malleus, that which is hammer shaped, that's why the name malleus has come. So, or it can also be called as hammer bone. Incus, anvil bone because of shape, anvil shape, and stapes, stirrup bone. So like this, malleus, hammer bone, incus, anvil bone, and stapes, stirrup bone. Then even they have asked what is their evolutionary origin also. So malleus is modified form of articular. Modified form of articular. Then incus, it is the modified form of quadrate. So it is the modified quadrate. It is the modified quadrate. And whereas stapes, it is the modified hyomandibula. So it is the modified form of hyomandibula. So it is the modified hyomandibula. So like this, there are three tiny bones which are present in the middle ear: malleus, incus, and stapes. Malleus, modification of articular; incus, modified quadrate, and stapes, it is modified by hyomandibula. So this is the evolutionary origin from where these bones have been developed. Okay, malleus from articular, incus from quadrate, and stapes from hyomandibula. So this is very important question. Name the three middle ear ossicles and what is their evolutionary origin. So the three tiny bones that which are present in the middle ear are only called as ear ossicles: malleus, incus, and stapes. Malleus, modified form of articular; incus, modified quadrate; and stapes is modified hyomandibula. Malleus, hammer bone; incus, anvil bone; and stapes is stirrup bone. So totally 5 important questions VSAQs we have learned from this chapter, that is musculoskeletal system. Already in excretion chapter also I have given 5 important VSAQs. So totally 10 VSAQs. Okay, so today you should learn. Even in yesterday's session also 11 VSAQs I have given from 3 chapters: digestion and absorption, breathing and exchange of gases, and then body fluids and circulation. Yesterday 11 and today again 10. So 21 questions, 21 VSAQs. And next we need to learn from neural control and coordination, endocrine system and their regulation. Okay. So let us go with neural control and coordination. Let me tell 2 more questions today in today's session, and remaining in the previous session, let us see. So from neural control and coordination, very, very important VSAQ is, what is meant by arbor vitae? What is meant by arbor vitae? Okay. So this is the question, what is meant by arbor vitae? So actually, you know that in brain, the gray matter is present in the cortex region, and whereas white matter is present in the, so gray matter, it is present in the cortex region, and white matter, It is present in the inner portion. So now arbor vitae, it is the inner white matter of cerebellum which projects out into a sheath of gray matter. So what is meant by arbor vitae? The inner white matter of cerebellum, that which projects out as a sheath of gray matter, that which projects out as a sheath of gray matter of cerebral cortex, so that it appears like a branching tree. That's why even in the brain diagram also, whenever we show cerebellum, we will draw like a tree, right? So it appears like a branching tree. So that branching tree appearance is only called as arbor vitae. And what is the meaning of that? It is the inner white matter of cerebellum which projects out into a sheath of gray matter, which projects out as a sheath of gray matter or into a sheath of gray matter, and it appears like a branching tree. So that is only called as arbor vitae. Then next, very important question. What is all or none principle? Very, very, very important question. What is all or none principle? So the action potentials that which are generated, the action potentials means what? Electrical currents or nerve impulses. So the action potentials that which are generated at threshold stimulus or supra-threshold stimulus. Threshold stimulus or supra-threshold stimulus, but not at sub-threshold stimulus. The action potentials that which are generated at threshold stimulus or supra-threshold stimulus, but not at sub-threshold stimulus, Okay. So that means either at supra-threshold or threshold, they will be generating the electrical currents, but it won't occur at sub-threshold stimulus, which means the nerve impulse is, so what is the meaning of that then? Which means the nerve impulse is either conducted totally or not conducted at all. So it would be conducted totally or not conducted at all. So that is only called as what? All or none principle. So the electrical currents that which are generated at threshold stimulus or supra-threshold stimulus, but the action potentials are the electrical currents, they won't be generated at sub-threshold stimulus. That means what is the meaning? Here the nerve impulses or the electrical currents, so now they will be conducted totally, or otherwise they won't be conducted at all. So that is only called as what? All or none principle. So the action potentials which is generated in response to a threshold stimulus or supra-threshold stimulus, but that doesn't occur at sub-threshold stimuli, which means that nerve impulse is either conducted totally or not conducted at all. So that is only called as all or none principle. So definitely you should learn these two questions. Definitely one will come. So arbor vitae is the inner white matter of cerebellum that which projects out into a sheath of gray matter, so which appears like a branching tree, is only called as arbor vitae. And all or none principle, action potentials that which are generated at threshold stimulus or supra-threshold stimulus. That means in response to threshold or supra-threshold stimulus, whatever the electrical currents are getting generated, but they are not generating at sub-threshold stimuli. So which means that these electrical currents or completely gets conducted or they won't be conducted at all. So that is only called as all or none principle. So that's all for today's session. Totally 12 important VSAQs I have taught you. So kindly learn today only. If you have any doubts, you can ask me. And already for LAQs and all recorded videos are there. For SAQs also, if you have any doubt, you can listen again and again by going into that. Okay. So if you have any doubts, you can ask me, those who have joined. Raise your hand so that we will unmute you. Yes, any doubts, children? How many join? Okay. Raise your hand. No doubts? They are not raising. Okay. So, no doubts for you. I can take it. Fine. That's all for today. And thank you, children.